Hi, welcome to today's class session. Today we are going to be discussing synaptic transmission, which is covered in Chapter 5. Just a reminder, make sure you're practicing labeling an action potential as shown here. This is included in the Chapter 4 video that I posted last week, but you should be able to label the action potential before your first exam, so make sure you understand this thoroughly. I also have a handout available on Canvas that you can print out so you can practice on your own. Today we are going to be discussing synaptic transmission. So first, I'm going to discuss the key differences between electrical synapses versus chemical synapses. And this is what allows neurons to communicate with one another. We're also going to be discussing major neurotransmitters, all the neurotransmitter types, how they're synthesized and stored in the neuron, and we're also going to be talking about exocytosis and the role that calcium plays in the removal of neurotransmitters at the synaptic cleft. Remember that we've already discussed that nerve cells have an excitable membrane, meaning that they can generate nerve impulses known as action potentials that can actually propagate down the axon. They do not diminish over time, and they can occur on long sensory nerves, sometimes up to a meter long. So for this information to be processed to the rest of the nervous system, for example, all the way from the brain down to a muscle in your foot, perhaps, these neural signals must be passed on to other neurons. So we're going to talk about the different modes of transmission. This is known as synaptic transmission. By the end of the 19th century, it was recognized that this transfer of information from one neuron to another occurs at a specialized site of contact. An English physiologist, Charles Sherrington, gave the name synapse to this area. Remember that the purple neuron here is known as the presynaptic neuron, whereas this orange neuron is known as the postsynaptic neuron. And it's this area known as the synapse that's the specialized area in which neurotransmitters can be sent from the presynaptic neuron to the postsynaptic neuron. And if you remember, we've talked about interneurons, which are the most typical type of neuron in which they're communicating with one neuron to the next. Other neurons can also communicate with other cells, such as muscle cells, but this is the most typical form of synaptic transmission. And remember that information is generally going to flow in one direction from one neuron to the next, so from the presynaptic cell to the postsynaptic cell, and that's the target cell. And this transfer of information is known as synaptic transmission. Synaptic transmission is necessary for life and function, and synaptic transmission is simply the chemical conversion of the electrical signal from the action potential. In our last class, we discussed how action potentials typically occur here at the axon hillock, and this is due to a high volume of sodium ion channels that sit at the axon hillock. When extracellular sodium enters here, it can cause the cell to become depolarized, meaning the inside of the neuron becomes more positive at rest compared to its negative resting membrane potential. When the cell becomes depolarized, it generates this nerve impulse or action potential, where the action potential can jump down from node to node along the axon proper. This is known as the nodes of Ranvier here in between the myelin sheath. And the action potential will then come to the axon terminal, where this electrical signal can then become a chemical form of transmission. And so here we have neurotransmitters that sit inside of the synaptic vesicles, and these vesicles, remember, I've defined them as these boats, sort of, and the boats can come down to the synaptic cleft with the neurotransmitters, and we're going to talk later about how exocytosis can cause the release of these neurotransmitters from the synaptic vesicles, thus entering the synaptic cleft to communicate with the postsynaptic neuron. So synaptic transmission is a very large and fascinating topic, and the actions of either psychoactive drugs or mental health disorders and simply the neural basis of learning and memory is all necessary and cannot be performed without synaptic transmission. So, is the signal transmitted in a synapse electrical or chemical? For a long time, there was a large debate whether synapses communicated via electrical or chemical activity, but the answer is both. We have both electrical and chemical synapses in our nervous system. And we're mostly going to talk about chemical synapses, but we will talk about one kind of electrical synapse called the gap junction, which is just less common than a typical chemical synapse. Chemical synapses are actually more unreliable because they run the risk of becoming blocked. Later in today's class, we're going to be discussing something called an antagonist, and this is when an antagonist can come and block a particular channel on a neuron, which can affect chemical transmission. Electrical synapses have a far less risk of becoming blocked, but if they do, it can have very large consequences. Thus, there are times when neurons need to utilize electrical transmission or chemical transmission, and we're going to discuss the key differences between the two types. First, I want you to know that chemical synapses occur when neurotransmitters are released from the synaptic vesicles into the synaptic cleft. So this is an important mode of information transfer between the cells, whereas electrical synapses are actually the direct transfer of ionic current, and there are fewer of these types of synapses in the nervous system. Chemical synapses require ligand-gated ion channels on the postsynaptic neuron to transfer neurotransmitters from the presynaptic cell to the postsynaptic cell, whereas electrical synapses require something called gap junction channels that are only about 3 nanometers apart to transfer ions from cell to cell. And these ions can actually transfer from the cytoplasm of the presynaptic neuron to the cytoplasm of the postsynaptic neuron. Interestingly, these gap junctions are very small. They're only 3 nanometers apart, and sometimes this is even difficult to see with an electron microscope. If you remember, we've talked about the typical synapse of neurons and how the synaptic cleft is generally about 20 nanometers. So thus, electrical synapses are much smaller and have these tighter gap junctions. Chemical synapses are not bidirectional, meaning they only move from the presynaptic neuron to the postsynaptic neuron. This is how information is transferred from cell to cell, whereas the electrical synapses can be bidirectional. This is when the ionic current can be passed between the presynaptic neuron to the postsynaptic neuron. This generates something called postsynaptic potentials, which we're going to discuss later on. And eventually, these postsynaptic potentials can actually become large enough to initiate another action potential that can be sent from the postsynaptic neuron to the presynaptic neuron. So this is largely a key difference between these two types of synaptic transmission. So my main takeaway from this slide is that chemical synapses are not bidirectional. They can only move from the presynaptic neuron to the postsynaptic neuron, whereas electrical synapses can be bidirectional, in which they can move back and forth from the presynaptic to the postsynaptic neuron, and then again from the postsynaptic neuron to the presynaptic neuron. And we're going to discuss this more in depth throughout today's lecture. So here is the chemical versus the electrical synapse. I've provided this handout on Canvas for y'all, so you can print this out to help you with note-taking or just to get a better visual, but we're going to go through each one. First, we're going to talk about the electrical synapse just because it's a more simple synapse and it's easier to understand before we get into a more complicated chemical synapse. Action potentials are actually required to stimulate both chemical and electrical synapses. So I think this can be confusing to students sometimes, so I want to make this abundantly clear that although chemical synapses are named chemical, they also have electrical properties because an action potential is required to stimulate this form of chemical synaptic transmission. So both synapses are utilizing action potentials to generate transmission. Now we're going to go more in depth in the next couple of slides. First, let's talk about electrical synapses. The existence of electrical synapses was actually proven in the late 1950s. And interestingly, this makes sense because the electron microscope was invented in the 1950s. We now know that the electrical synapses are common in the brains of invertebrates, vertebrates, and mammals. Electrical synapses allow the direct transfer of ions from the cytoplasm of one neuron to the next. This can even include epithelial cells, smooth and cardiac muscle cells, liver cells, and glial cells. So electrical synapses may not just occur between neuron to neuron. They can also transfer this ionic current to other cells around it. So interestingly, electrical synapses occur at these specialized sites known as a gap junction. And we're going to talk more about how the gap junction develops. Gap junctions occur between cells in nearly every part of the body, so they interconnect many non-neural cells as well. Gap junctions between neurons and other cells are actually really common during early brain development. So evidence suggests that during prenatal and postnatal brain development, gap junctions allow neighboring cells to share both electrical and chemical signals that may help coordinate their growth and maturation. Later on in this class, we're going to discuss how the brain develops and how the entire nervous system actually develops from one flat sheet of cells. This happens 17 days after conception. We're going to talk about how important this process is to create the entire nervous system and how electrical synapses are playing a role in helping cells get to where they need to go. This is called neuronal migration. When gap junctions interconnect neurons, they're actually known as electrical synapses. And so gap junctions, as I noted previously, are also found in between cells that usually migrate together during development. And that's why they play such an important role in the developing nervous system. You can actually inject dye into one of these cells, and you can see how it will spread into all of its connected cells around it. Gap junctions between neurons are electrical in nature, and they are about 3 nanometers apart. This gap is extremely small, and remember, at a chemical synapse, that gap is about 20 nanometers. So both of the chemical and electrical synapses can't actually be seen without the use of the electron microscope. If you remember from previous lectures, we've talked about how a light microscope or a compound microscope can't be used to look at these gaps. It is required by an electron microscope. This narrow gap is connected by proteins, and these proteins are known as connexins. So connexins are these small subunits or 3D structures. We've talked about subunits before when we were discussing the tertiary structure of proteins. Remember, there are several different types of protein structures you're going to need to know for your first exam. The primary structure, which is simply a single strand of amino acids that come together. Remember that these amino acids can come together to form a helix structure, which is the secondary type of protein structure. And then this third protein structure is known as the tertiary structure, and this develops into a 3D shape. These 3D shapes are called subunits, and the connexin subunits, which is a particular type of protein to create gap junctions, can come together to form something called a connexon. I like to think of the connexin as a small Lego piece that can come together with other sets of Lego pieces to form one structure. The connexons that form this larger structure can come together because we have the top connexon and the bottom connexon, and these form the gap junction. The gap junction is simply a pore or channel that allows ions to come inside into the presynaptic neuron and then into the postsynaptic neuron. And, as I previously noted, these synapses can be bidirectional. So an electrical synapse can also send back information, which is ionic current, from the postsynaptic neuron back to the presynaptic neuron. Two connexons that come together are what form the connexon, and this is our gap junction or channel where ions pass through. The channel allows ions to pass directly from the cytoplasm of the presynaptic neuron to the postsynaptic neuron, or in a bidirectional manner from the cytoplasm of the postsynaptic neuron back to the cytoplasm of the presynaptic neuron. So this is very different from a chemical synapse. When we discuss chemical synapses later on in today's lecture, we'll understand that they are not bidirectional like the electrical synapse. So that makes this type of synapse very special. Here's a look at the gap junction in between two neurons. Gap junction channels occur in the phospholipid membrane, just like we've talked about before with ion channels and sodium-potassium pump that can also sit in the phospholipid membrane. Gap junction channels have to be very close together. They're only 3 nanometers apart. And they sit right inside of the phospholipid bilayer. Remember, we've talked about the phospholipid bilayer, and you will need to know this for your exam. The hydrophilic heads and the hydrophobic tails are what compose this important membrane that help prevent any ion or chemical from getting inside of the cell unless it's supposed to be there. Gap junction channels exist here, and they allow the direct ionic current from one cell in which the ion can come into the channel, as shown with my laser pointer, come through the channel through this pore, and back into the cytoplasm of the postsynaptic neuron. First, we're going to look at electrical transmission. This is mediated by clusters of intracellular channels, which we know as gap junction channels, shown here in gray. Gap junction channels sit in between the two cells, and the two cells I'm using in this example are a presynaptic neuron and a postsynaptic neuron. Remember, this can occur between neurons and other types of cells, but in this example, I'll be discussing interneurons, in which neurons communicate with other neurons around them. This space is extremely small. It's only about 3 nanometers in size, and so thus this allows the direct current of ions to flow from one cell to the next. First, an action potential in the presynaptic neuron needs to occur to generate a small amount of ionic current. Ions can directly flow from the cytoplasm of the presynaptic neuron through the gap junction channel to the cytoplasm of the postsynaptic neuron. This direct flow of ions from the presynaptic cell to the postsynaptic cell can generate enough electrical current to induce something called a postsynaptic potential, or PSP, in the postsynaptic neuron. Interestingly, most electrical synapses are actually bidirectional. So when the postsynaptic neuron generates an action potential from this excess ionic current or electrical current, it can actually induce a PSP in the first neuron. So ultimately, my biggest takeaway for this slide is I want you to understand that electrical synapses can actually directly send ions from the presynaptic cell to the postsynaptic cell, and in a bidirectional manner from the postsynaptic cell to the presynaptic cell. Both of these are utilizing action potentials to induce this current. Let's take a better look at postsynaptic potentials, also known as PSPs. So in this example, we're also going to be looking at two neurons that are connected via this gap junction to induce electrical synaptic transmission. So here we have our presynaptic neuron that's trying to communicate with the postsynaptic neuron below it. In image A right here, this is an actual picture that was taken from an electron microscope of two neurons communicating. And as you can see, the gap junction is incredibly small, allowing this direct flow of ions from the cytoplasm of each cell to the next. So the postsynaptic potential that's generated by a single electrical synapse is actually very small and usually doesn't trigger an action potential on its own. However, there's this common notion in neuroscience that cells that fire together wire together. If we have several neurons that are sitting around each other that are generating postsynaptic potentials, this can induce an action potential. So essentially, several PSPs that are occurring simultaneously may strongly excite a neuron, generating an action potential in which that neuron is going to become depolarized, and extracellular sodium will rush into the cell and generate an action potential. Thus, this relationship between the two neurons is bidirectional, because if an action potential occurs and induces ionic current in the postsynaptic neuron, it can send this current back to the presynaptic neuron. So generally, an electrical synapse is bidirectional, in which these two neurons can be signaling via ion current between each cell. If you want to take a better look at synaptic transmission, this video is a great demonstration of that. I believe it was created by Harvard University, and they created this 3D model to show us what electrical and chemical synapses look like in between two neurons. I'm going to post a link for this in the module for Chapter 5, so go ahead and take a look at this if you want to visualize an electrical synapse or a chemical synapse. Now we're going to discuss the prototypical chemical synapse. So this is a different type of synaptic transmission from the previously discussed electrical synapse. Here's a look at our chemical synapse. So in this example, we're also going to be looking at two neurons that are communicating. Remember, two neurons that communicate together are called interneurons. Here we have a presynaptic neuron and a postsynaptic neuron. Remember, a chemical synapse is a little more complicated, and it does not have a bidirectional relationship. So thus, the presynaptic neuron will be sending signals to the postsynaptic neuron, but the postsynaptic neuron will not send signals back to the presynaptic neuron. So this is different from the electrical synapse that we just talked about. But the presynaptic neuron can become depolarized, meaning that extracellular sodium can rush into the cell, causing an action potential to propagate down the axon proper. Remember, this typically begins at the axon hillock, in which the action potential itself jumps from node to node at the nodes of Ranvier on the myelin sheath until it reaches the axon terminal. When it reaches the axon terminal, action potentials can actually induce the opening of voltage-gated calcium channels. The activation of these calcium channels allows calcium to rush into the neuron, and calcium performs something known as exocytosis. This is the release of neurotransmitters from inside synaptic vesicles. Now, if you remember, I've talked about how synaptic vesicles function like boats. They carry the neurotransmitters from the axon hillock down to the axon terminal. It's also important to note, and this is good review for you guys, that anterograde or retrograde transport plays a key role in getting these synaptic vesicles down to the axon terminal. If you remember, we've talked about anterograde transport, in which synaptic vesicles carrying neurotransmitters are carried from the axon hillock down to the axon terminal. And remember, there's a specialized type of protein known as kinesin, and kinesin is a motor protein that will actually carry the synaptic vesicle down the microtubules of the axon proper. Interestingly, once these synaptic vesicles reach the axon terminal, calcium is necessary to remove the neurotransmitters from the synaptic vesicle so that they can come into the synaptic cleft. Remember, the synaptic cleft here is the space in between neurons, and in a chemical synapse, this is about 20 nanometers in size. Once neurotransmitters reach the synaptic cleft, as shown here in red, they can be received by the postsynaptic neuron. The postsynaptic neuron will most likely have specialized ligand-gated ion channels. If you remember, ligand-gated ion channels are specialized receptors in which a neurotransmitter can bind to that receptor, and if it's the particular neurotransmitter that will allow the opening of that ion channel, the gates will open. This is also known as conformational change, when its protein structure can change shape to open the ion gates, and the neurotransmitter and other ions can potentially go inside of the cell. So for example, let's think about glutamate. Glutamate is our main excitatory neurotransmitter, and it utilizes a very special receptor known as the NMDA receptor. The NMDA receptor is the main glutamate receptor in the brain, and we're going to talk about it in more depth in our next lecture. If the neurotransmitter glutamate comes and binds to the NMDA receptor, it's going to allow the ion channel gates to open and allow neurotransmitters to pass into the next cell. When this occurs, membrane potential can change because the binding of this ligand, which is glutamate, can induce the flow of ions into the postsynaptic cell. This could potentially generate another action potential. It can also change gene expression or induce other biological cascades that we're not going to go into depth about. So here, the key thing I want you to understand is that we have ligand-gated ion channels here, and a specialized neurotransmitter, such as glutamate, can come and bind to that channel, which will induce conformational change or the opening of these ion channel gates and allow ions to come inside of the postsynaptic cell. And if this ion perchance could be sodium, this could cause depolarization of the cell, which will cause it to become more positive and maybe generate another action potential. So thus, action potentials are not only necessary for electrical synapses, they're also necessary to induce chemical synapses. And that is my key takeaway for this slide. Here's another look at our chemical synapse. So as you can see, a chemical synapse generally begins here at the axon hillock. We typically have concentrated amounts of ion channels. So if we had extracellular sodium present, it could come into the cell and depolarize it, meaning it would become more positive than it is during its resting state, which will induce the action potential. And remember, you should be reviewing the action potential for your exam. You need to know the different steps, which is depolarization, the overshoot, where the absolute refractory period occurs, the repolarization of the cell in which it becomes more negative again due to an efflux of potassium, and hyperpolarization, where the cell can sometimes become more negative than it is during its resting state. This entire process occurs along the axon, and as the action potential propagates down the axon proper, it does not diminish in frequency. Once it reaches the axon terminal, this can induce the opening of calcium ion channels, and calcium can come inside of the cell and allow synaptic vesicles to release neurotransmitters as shown here. These little blue bubbles here are synaptic vesicles, and when these neurotransmitters enter into the synaptic cleft, they can be taken up by receptors in the postsynaptic cell. If you remember, we've also talked about different types of synapses. If you remember, there are axodendritic synapses, and so this would be a great example of that, because an axon of one neuron is communicating with the dendrite of another neuron, and this would be known as axodendritic. So it wasn't until the year of 1921 when Otto Loewi actually discovered the very first neurotransmitter, and the discovery of the first neurotransmitter, which is acetylcholine, provided more support for chemical synapses. Remember, at this time, the electron microscope was not invented, so it was still difficult to look at synaptic transmission. So Otto Loewi had to be very creative. He performed an experiment, and this experiment showed that electrical stimulation of axons innervating a frog's heart could cause the release of a chemical, which we now know as acetylcholine, that could mimic the effects of neuron stimulation on the heartbeat. Now, we're going to talk about this experiment and how he performed it. This experiment has a really fascinating backstory. So Otto Loewi was a scientist who was trying to understand how neurons communicated. He knew that neurons must be sending some type of chemical to one another to transmit information and communicate. But at this time, neurotransmitters hadn't been discovered. So this is the question that he was trying to answer. What type of chemical is utilized by cells to help them communicate? So what's really, really cool about this experiment is he actually thought of it in a dream. One night he had a dream about how he could test his theory, but he fell back asleep and he couldn't remember the experiment. He was really frustrated by this, and another night he went back to sleep and he had the dream again, he actually woke up in the middle of the night and went straight to his lab to perform the experiment so he wouldn't forget it. And I believe this was actually on Easter morning, so it's pretty interesting. But what he did when he got to his lab is he took one heart from a frog, and it was still connected to its vagus nerve. Here in orange, this is symbolizing the vagus nerve of the frog heart. Now, the vagus nerve is a very crucial cranial nerve that's known as the wandering nerve. The vagus nerve has axons that project into many systems of the body, all the way from the brain to the gastrointestinal tract, which is super important for communication between the gut and the brain, which we'll talk about later on in this class. But right now, I just want you to understand that the vagus nerve innervates important areas like the gastrointestinal system and the heart. So the vagus nerve allows communication from nerves in the neuromuscular junction of the heart back to the brain. And so essentially, this first heart was placed in a saline solution, so it's just that salty extracellular, mimicking the salty extracellular fluid of the cell. And this heart was still connected to its vagus nerve, and it was connected to a stimulating device. Additionally, the second frog heart was placed in a saline solution as well, but its vagus nerve was not connected, and it was not electrically stimulated. Only the first heart would receive electrical stimulation. But there was an area where fluid could transfer from the first frog heart to the second frog heart. So, Louis, what he did was he stimulated the first frog heart with electricity. And when he stimulated the first heart via the vagus nerve, he found that it caused the heartbeat to slow down. So interestingly, after heart number 1 started to slow down, the second heart also slowed down. As you can see, this is the rate of heartbeats, and both hearts started to slow after stimulation of heart number 1 via the vagus nerve. So how is this occurring? Interestingly, he found that electrical stimulation of the vagus nerve released the chemical or neurotransmitter that we know today as acetylcholine. He called this Vagisstoff because he was German, which literally means like a vagal fluid. And so he thought this chemical from the vagus nerve was inducing the slowed heart rate in heart number 1, and that this Vagisstoff substance, which we now know as acetylcholine, was transferred to heart number 2 to slow down the heartbeat. Interestingly, stimulating the vagus nerve calmed down the heart and slowed it down. This Vagisstoff substance, which is literally vagus substance, is known as acetylcholine, and this is the very first neurotransmitter that was ever discovered. We now know today that acetylcholine plays a really important role in our parasympathetic nervous system response. We haven't talked about different subtypes of the nervous system yet, but this is a subtype of the peripheral nervous system that plays an important role in allowing our body to rest and digest. So stimulation of the vagus nerve to the heart increased acetylcholine, and acetylcholine occurs at the synapse between nerve cells and skeletal muscle. So interestingly, acetylcholine caused the heart to slow down, and that's why we saw this change in heart rate in heart number 1, which eventually allowed the heart number 2 to slow down its heart rate as well, because the fluid transfer of acetylcholine from heart number 1 was then brought to heart number 2. And unlike at the heart, if acetylcholine's released at the skeletal muscle, it can actually cause excitation and contraction. So acetylcholine can play different roles, and it's very important for maintaining our heart rate and our heartbeat. And here's a look at a real-life synapse. This was taken with an electron microscope. So here we can see the presynaptic neuron, and at the bottom here we can see the postsynaptic neuron. And this is a very small space in between our two cells, which is only about 20 nanometers in size. Chemical synapses can occur between different types of cells. So first, the most common is neuron to neuron. This is the majority of connections in the nervous system. Remember, neurons that communicate with other neurons are known as interneurons. But neurons can also communicate with muscle cells. This is one of the most studied types of connections, and this occurs at the neuromuscular junction. These are really accessible for researchers because they're out in the periphery of the body as opposed to opening up the brain. So this is a way that we can actually study how neurons can communicate with muscle cells. Now we're going to talk about neuron to neuron. So this should be review for all of you. We discussed the different types of synapses in Chapter 2 already, but I want to briefly review it again because it will come up on Exam 1. So in the central nervous system, there are different types of synapses which can distinguish which part of the neuron is postsynaptic to the axon terminal. So here in image A, we see that this axon is going to be synapsing onto the dendrite of the postsynaptic cell. This is known as axodendritic, and this is one of the most common types of synapses. We also know that the axon may synapse onto the soma of the postsynaptic neuron, and this is known as axosomatic synapses. This is likely to inhibit the activity of other neurons. In some cases, the postsynaptic membrane is on another axon, and remember, these are known as axoaxonic synapses, and they also typically inhibit the activity of other neurons. There's also something known as dendrodendritic synapses, in which these are typically found in the olfactory bulb, which is an area of the brain that's very helpful for differentiating different smells. They're also found in the retina, in the color contrast processes, but essentially these are dendrites that synapse onto other dendrites. So these are the different types of synapses I want you to know, and hopefully this should be review for y'all since we covered it in Chapter 2. Similarly to the way that we can categorize different types of neurons, we can also categorize different types of synapses based on their shape. So here are two examples. They're known as Gray's type 1 and type 2 synapses. First, we're going to focus on Gray's type 1 synapses shown here. So if we look at image A, we know that Gray's type 1 synapses typically have round vesicles. Remember, these synaptic vesicles are carrying neurotransmitters. Additionally, they usually have an asymmetrical membrane, and they usually will generate excitatory processes, meaning this will induce excitation in the postsynaptic neuron. A great example here is if the neurotransmitter glutamate was being dumped out into the synaptic cleft, it could generate excitation in the postsynaptic cell. So glutamate could potentially be carried in these round vesicles. Another example is Gray's type 2 synapses. And Gray's type 2 synapses typically have flattened vesicles, and they're typically symmetrical. This usually induces inhibition in the postsynaptic neuron. So a good example of this would also be the neurotransmitter GABA or glycine. Both of these neurotransmitters typically induce inhibition in the postsynaptic neuron. So thus, different types of neurotransmitters will be transferred in different types of synaptic vesicles, and the actual synapse itself can be different in shape depending on the type of neurotransmitter that it is sending via chemical synaptic transmission. Now we're going to discuss neuron to muscle synapses. Now I've used the example of the neuromuscular junction before when we were discussing the simple reflex. So now we're going to look at this in more depth. So here at the postsynaptic membrane, the motor end plate contains junctional folds with numerous neurotransmitter receptors. So thus, at the neuromuscular junction, we actually have nerve cells that can synapse onto these muscle fibers to generate movement. Neurons in the motor system synapse directly onto muscle, and that's why it is called the neuromuscular junction. These synapses typically have acetylcholinergic receptors. Thus, these are neurons that typically secrete acetylcholine. If you remember before, we've talked about how acetylcholine can play an important role at the neuromuscular junction, and we used this example when I was talking about Otto Louis' experiment. The neuromuscular junction is immensely critical in ensuring that we can move properly. Our nerve cells need to be able to synapse onto muscle fibers and perform properly in order for us to command motor movement and to prevent unwanted movement. Sometimes this is sadly disrupted, and an example of this is an autoimmune disease known as myasthenia gravis. This is a disease in which B cells release antibodies that bind to and block the acetylcholinergic receptors at the neuromuscular junction, and this causes muscular weakness. Just a quick refresh, B cells are really important immune cells in the adaptive immune response. We have two different types of immune responses. We have the innate immune response, which we are born with. This helps us fight off pathogens like bacteria that we come across every day. But we have a secondary immune response known as the adaptive immune response. And in this response, we have specialized immune cells known as B cells, and they essentially have this immunological memory. They can create antibodies based on viruses or pathogens they've seen before and create antibodies to protect us in the future. Sadly, in the case of autoimmune diseases, and in this example of myasthenia gravis, these antibodies can sometimes be detrimental in that they're attacking the body itself. It's almost recognizing this area of the body as a pathogen or a virus, and thus is actually inducing inflammation and muscular weakness. B cells block the normal activity of acetylcholine at the neuromuscular junction, and thus prevent acetylcholine from doing its job. That's what induces muscular weakness. Now let's look at an example of neurons to other synapses. For example, the hypothalamic neurosecretory neurons project from an area of the brain known as the hypothalamus. We're going to be discussing the hypothalamus in very large depth in this class because it is absolutely imperative for the hypothalamus to help us regulate homeostasis and the regulation and release of hormones and neurohormones. The hypothalamus, shown here in blue, has specialized cells, and their axons project down to the pituitary gland. The pituitary gland is composed of these two lobes, the posterior pituitary, shown here in purple, and the anterior pituitary, shown here in green. So these cells that live in the hypothalamus have axons that actually project down to the pituitary gland to help release important hormones such as sex hormones, stress hormones, and neurohormones. These cell bodies actually synapse onto the anterior and pituitary gland. So thus, this has different effects depending on where they synapse. So let's look at this in more depth. We have specialized neurons that live in the hypothalamus and project down to the anterior pituitary gland, shown here in green. The anterior pituitary is important for the release of hormones into the bloodstream, including growth hormones and sex hormones that have to do with lactation or perhaps puberty. Interestingly, the anterior pituitary is actually a gland itself, and it's actually not a brain structure, whereas the posterior pituitary is actually still part of the brain. But we need this connection between the hypothalamus and the synapsing of cells in the hypothalamus onto cells in the anterior pituitary in order to release important hormones into the bloodstream. That's why a tumor in this area could be very detrimental to your health. And we're going to talk about that later on when we get to chapter 15. The posterior pituitary is a part of the brain, and there are specialized neurons that live in the hypothalamus that synapse onto cells in the posterior pituitary. The posterior pituitary, shown here in purple, is really important for releasing neurohormones. There are two types of neurohormones we're going to discuss in this class, including vasopressin, which is also synonymous with antidiuretic hormone, and oxytocin. You've probably heard of both of these, but vasopressin helps with kidney function and water retention, and it helps decrease the amount of urine that we produce. Oxytocin is the love hormone. It's the hormone that helps us bond. For example, skin on skin contact between a mother and her newborn child releases oxytocin, in addition to other bonding activities like sex or cuddling. So, interestingly, these neurohormones will only be released if there's synapsing occurring from neurons in the hypothalamus down to cells in the posterior pituitary. And this is why synapses at the chemical level are so important. Interestingly, we also know that cholinergic neurons can synapse onto other areas like the spleen. This is an image of a mouse spleen, and there are cholinergic neurons inside of it. The spleen is innervated with the splenic nerve, and this communicates directly with the vagus nerve. If you remember, we've already discussed the vagus nerve or wandering nerve, because the vagus nerve innervates several different areas of the body and allows neural communication from the brain to other organs, such as the spleen. If you remember, we've already discussed the vagus nerve in the context of Otto Loewi's experiment, in which he used electrical stimulation of the vagus nerve to increase synaptic transmission at the frog heart. Cholinergic neurons can actually synapse onto the spleen and regulate inflammatory activity in macrophages in the spleen. I've briefly discussed macrophages before when we talked about microglial cells in Chapter 2. If you remember, microglial cells function as the brain's resident immune cell because they can perform phagocytosis when bacteria or pathogens potentially enter bloodstream to the brain. Any type of inflammatory trigger can activate macrophages and microglial cells. Macrophages are these important immune cells that perform phagocytosis in the periphery of the body. For example, if you had gram-negative bacteria or a pathogen in your blood, macrophages would become activated to eat up those pathogens and rid them from your body. So interestingly, cholinergic neurons can synapse here and help regulate that inflammatory activity and macrophages in the spleen. This neural immune signaling actually helps to regulate the immune system response and lymphocyte cell function. And throughout this class, I'm going to be talking about the immune system a lot because it's actually largely connected to our brain health. And we need to ensure that we have regular levels, basal levels of inflammation, and a proper activation of our immune system so that it doesn't induce inflammatory diseases like arthritis, dementia, Alzheimer's disease, and even cardiovascular disease. Now we're going to discuss neurotransmitters. So what are neurotransmitters exactly? These are small chemical messages that are produced by our cells, and they can be transmitted from cell to cell. Now we've talked about neurotransmitters quite a bit, but we haven't talked about the different types of neurotransmitters in depth. We know that there are several different types. So far, we've discovered more than 60 different types of neurotransmitters. And they're endogenous, so they're actually produced inside of the neuron itself. So here's a look at neurotransmitters, and we've seen a picture like this in many of my previous slides. We know when a cell is activated by an action potential, neurotransmitters are released into the synapse, which is also known as the synaptic cleft. And so these neurotransmitters that are carried in synaptic vesicles can be removed from the synaptic vesicles when calcium rushes into the cell. Calcium performs something known as exocytosis, and this allows neurotransmitter molecules to come into the synaptic cleft and then bind to the postsynaptic neuron. Remember that specific receptors on neighboring cells like this postsynaptic neuron are the ones that take up the neurotransmitters for chemical neurotransmission to occur.